Hello, everyone, and welcome to today's live broadcast, Anchored Protein Kinase-A, PKA, as Gatekeeper for Connexin-43 Gap Junction, presented by Dr. Guillaume Pidoux, Senior Researcher at NCMM-UMRS 1180. I am Xavier Gutierrez of LabRoots, and I will be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots and sponsored by Thermo Fisher Scientific. For more information about our sponsor, visit www.thermofisher.com. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the Ask a Question box located on the far left of your screen and type your questions into the drop-down box that appears on the screen. We'll answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, please use the Ask a Question box to let us know that you're having a problem. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located in the top right corner of the presentation window and follow the process to obtain your credits. Now, please join me in welcoming Dr. Pidoux. I will turn the presentation over to him.
Hi, my name is Guillaume Pidoux, and I'm Senior Researcher at INSERM, the French National Institute for Health and Medical Research at University Paris Descartes. And I will talk today and present some techniques and tools that we have been using to study protein-protein interaction and how, by using these tools, we could show that the anchoring of PKA can serve as a gatekeeper for Connexin-43 gap junction. But let's start with some background. With the cyclic AMP signaling, when you have a ligand on hormones that bound to the GPCR, the GPCR can activate adenine cyclase, which can produce cyclic AMP from ATP. Cyclic AMP can have many down effectors, such as the CNGs or EPAC, but the main down effector is Protein Kinase-A, or PKA. PKA is composed by two regulatory subunits and two catalytic subunits. Once you have cyclic AMP produced and bound to the regulatory subunit, it releases the catalytic subunits, which can turn active and can phosphorylate serine and threonines. It exists two types of PKA. PKA type 1, which is mostly but not exclusively cytosolic, and PKA type 2, which is mainly anchored. You can regulate in space and time the activity of this protein kinase-A. In time, by acting directly to the diamylate cyclase, by decreasing the production of cyclic AMP, or directly by activating phosphodiesterase, that's our enzyme, which hydrolyzes this cyclic AMP. And in space, by family of protein that we call AKAPs that are able to anchor protein kinase-A specifically into a cell. In the human placenta is a very important organ during the pregnancy. It will provide all the nutrients to the fetus for the fetal growth and will help the mother as well to the upcoming babies. So the structural and functional unit of the human placenta is the chorionic villus. It looks like a tree, and it baths directly into the maternal blood. On the right, you could see a schematic drawing of this chorionic villus. It's composed by a stromal core that possesses the fetal capillaries, and on this stromal core are, in purple, the villus cytotrophoblasts that are able to fuse together and form the external layer of this villus called the syncytiotrophoblast. And this syncytiotrophoblast is in contact direct with the maternal blood that races against the maternal arteries. This syncytiotrophoblast is the key structure of the human placenta. It will be responsible for all the feto-maternal exchanges between the mom and the fetus, providing all the nutrients. But it will be also the seats for all the production and the secretion of all the hormones of the pregnancy that will help the mother to prepare the upcoming babies, but as well to help for the fetal growth. It's possible to purify the cytotrophoblast from the human placenta, and these cells in vitro are able to aggregate between 24 and 48 hours of culture and then fuse to form a syncytial trophoblast after 72 hours of culture. On the picture, you can see trophoblastic cells where the nucleus was stained in blue with DAPI and the membrane in green by using an antibody raised against the desmoplaskin. So at 24 hours of culture, you could see an aggregation. At 72 hours of culture, you could see a large cytoplasm area in which you have nuclei stick together. This is the syncytium. This functional differentiation is also accompanied by an increase of production of hormone of the pregnancy, such as HPL for human placental lactogen and ACG for human chorionic gonadotrophin. Increase that is associated with the apparition of the syncytial trophoblast. Without cell fusion, there is no syncytial formation and no pregnancy at all. For many years now, we have been studied this fusion process, and we have found that many effectors can help the fusion. For instance, the EGF and the receptor, or the ACG and the receptor, initiate the process of fusion. This is what we call the competence stage. So ACG is also produced by the syncytial trophoblast and acts as an autocrine parotid loop to help the fusion of the cytotrophoblast. Then we also find some proteins such as CETO1 or the gap junction protein Connexin-43 that play a role in the cellular aggregation, that is the committant stage. And finally, the lipid bilayer imaging is in charge by syncytins. So we wanted to know if the PKA signaling is involved in the cell fusion process. And for that, we treated the cells with ACG or ACG and an antagonist of the Protein Kinase-A signaling that we call the H89. When we treated the cell with ACG, we could see that there is already, after 24 hours of culture, the presence of the syncytium that we don't have in the control. This is summarized on the histograms, where you could see that there is an increase in the percentage of fusion by the fusion index. But once we treated the cells with ACG and H89 that block the cyclic AMP PKA signaling, then we can observe presence of aggregation already and still at 72 hours of culture. That's also summarized on the histogram on the right. At 72 hours of culture, you could see a decrease in level of fusion index. So this indicates that the PKA signaling induced trophoblast fusion. Like I said earlier, you can control in space the protein kinase-A activation by anchoring the regulatory subunit to a family of proteins that we call AKAPs for A-Kinase Anchoring Protein. This is a large family of proteins that don't share any homology of structure, but they share 4 points that need to be present in this family. First, they have all to contain a PKA anchoring domain that we call A-Kinase Anchoring Domain. Then they also have to contain a unique subcellular targeting domain. So these AKAPs have to be localized into the nucleus, mitochondria, or endosome, for instance, or other proteins. They also have to present the ability to form a complex with other signaling molecules, such as phosphodiesterase, phosphatases, or other kinases. And finally, they should directly bind to or co-localize with specific PKA substrates in order to favorize the phosphorylation quickly of the target by the catalytic subunit. And this will provide a biological effect that you can observe. It's possible to dissociate the anchoring of PKA from the AKAPs by using anchoring protein disrupting peptides, or PPI, that will compete the anchoring of the regulatory subunit through AKAPs. It exists different types of PPIs. The type 1 specific anchoring disrupting peptides called RIAD. We also have super AKPS, which is a specific type 2 anchoring disrupting peptides, and some peptides that dissociate PKA type 1 and type 2 called HT31. So we we culture our cells with either RIAD or super AKAPs that dissociate the anchoring of PKA type 1 and type 2, or the scramble control, and treated the cells with ACG to promote cell fusion. And what we could observe is when we treated the cells with RIAD or super AKAPs, we then decreased the level of fusion compared to the scramble treated cells. But this indicates that anchoring of PKA through AKAPs controls trophoblast cell fusion. So we wanted next to identify the expression of AKAPs expressed into trophoblast. And for this, we decided to develop a mass spectrometry analysis assay. So we generate P-destination vector encoding the regulatory subunit type 1 or type 2 with a flag tag into a plasmid that will be transfected into E. coli BL21. We generate this recombinant protein and purify this PKR flag protein by using cyclic AMP agarose beads. We detach the beads from the recombinant protein by using excess of cyclic AMP. And then the recombinant protein were mixed with lysets from either a pool of cytotrophoblast or a pool of syncytial trophoblast in order to fish AKAPs expressed in both cell types. Then we came with an anti-flag magnetic beads to enrich the complex PKR and AKAPs, and did trypsination, and all the peptides were sent to LTQ orbitrap for AKAP identification. And we found many AKAP expressed into the cells, either in cytotrophoblast or syncytial trophoblast, AKAP specific for type 1 and type 2, and some AKAPs that are expressed specific for type 1 and type 2 that we call dual AKAP. So in order to decipher which AKAPs are involved into the control of cell fusion, we decided to realize an AKAP screening by using sRNA. So we designed sRNA for the AKAPs that have been found in the mass spectrometry analysis. And by using qPCR, we are able to measure the level of knockdown by using this sRNA. And in the same culture, we can measure also the impact of this sRNA on the fusion index. And what we could observe is that cells transfected with Ezrin sRNA present the highest level of decrease in cell fusion. So that indicates that this protein could be interested into the control of cell fusion. So what Ezrin is? Ezrin is a non-AKAP. It's an ERM protein, the same as rhodixin and myosin. It contains a FRM domain that's linked to other proteins, such as EBP50 or other exchangers, for instance. You have alpha helical domain. This is a central domain that linked to PKA type 1 and type 2, and the C-terminal domain that was known until now to link to actin. So what we did, we treated the cells with Ezrin sRNA or the scramble, and transfected the cell with lipofectamine 2000 CDs, and then we could see there is decrease in the expression of Ezrin compared to the scramble, and this is also associated with a decrease in the level of fusion, as we could see aggregation cells and decrease in the histogram. But then we wanted to reconstitute the fusion, so we co-transfected the cell with Ezrin sRNA and a plasmid that will generate a GFP Ezrin sRNA insensitive. And in that case, we were able to reintroduce the cell fusion that you can see the apparition of the green syncytium. So then we generate another P-destination vector that will produce a GFP Ezrin sRNA insensitive, in which we make mutation into the Protein Kinase-A domain. So this construct does not present the possibility to bind PKA. And in that case, we are not able to rescue the cell fusion process, and we found green aggregated cells. So this means that the PK anchored to Ezrin promotes cell fusion. Beyond all the proteins that we found in the laboratory to be involved in the control of of the cell fusion, we found that Connexin-43 to be important in the trophoblast cell fusion and differentiation. gap junction Connexin-43 is expressed in the trophoblast, and you need 6 connexin to form a connexin. And 2 connexins can dock each other to form a gap junction to allow cell-to-cell communication. Cell communication promotes cell fusion in our trophoblast, and this connexin can be phosphorylated by many kinases, such as PKA, PKC, or ERK. The PKA phosphatides are located into the amino acid 364 and 373 of the connexins, and it allows transfer of small molecules. So in order to see if connexin and Ezrin are able to form a complex together, we decided to realize co-immunoprecipitation by using dynabeads on which we cross-linked the antibody by using BF3. And when we co-immunoprecipitated Ezrin, we were able to find Connexin-43, but as well as PKA type 1 and type 2, and the catalytic subunit. We use AKAP-18, which is a known PKA type 2 AKAP, as negative control, and didn't find AKAP-18 to be in the same complex as Ezrin. Interestingly, if we did co-immunoprecipitation with Connexin-43, we're able to find Ezrin, PKA type 1 and type 2, and not AKAP-18. Then, if we realize a PKA catalytic subunit co-immunoprecipitation, we're able to find connexin, Ezrin, both PKAs, and of course, in this time, we found AKAP-18 to be associated with PKA. Again, if we did the PKA type 1 immunoprecipitation with the regulatory subunit, we found Connexin-43 to be together with PKA type 1, Ezrin, the catalytic subunit of PKA, but not AKAP-18. And if we did a PKA regulatory 2 subunit co-immunoprecipitation, then we found Connexin-43, Ezrin, and the catalytic subunit as well as AKAP-18. So this suggests that it exists in the human placenta and trophoblastic cells a PKA-Ezrin connexin signaling complex. We wanted next to measure or to support this complex by using a proximity ligation assay and using the dualing strategy. This strategy allows us to quantify the proximity between 2 targets, 2 proteins in the cells by quantifying the generation of dots that indicate proximity in the 40 nanometer range. All this is working. We use specific primary antibody that targets our protein of interest. Then we use specific secondary antibody, and if these both proteins are in the range of 40 nanometers, then you can generate a signal that can be quantified by generating a dot. And this way you can observe here. All the white dots correspond to proximity of the protein of interest in the 40 nanometer range. Here you could see that Ezrin co-distributes with PKA type 1, PKA type 2, as well as Connexin-43 with PKA type 1 and type 2. And finally, Ezrin and connexin also co-localize together, while the AGG control didn't show any staining or dot generation. We know from before that Connexin-43 were expressed mainly in the cytotrophoblast to allow the cell-to-cell communication, and in a less extent in the syncytial trophoblast. So we were interested to see where Ezrin would be expressed in the human placenta. So we did immunohistochemistry. So here is like placenta that we make slice, and we stain it with different Ezrin antibody or cytokeratin 7, which is a specific marker for trophoblast. And we stain as well the nucleus in blue with DAPI. And what you could see is Ezrin is highly expressed in the syncytial trophoblast layer, mostly highly at the apical pole, which is in contact with the maternal blood, but also at the cytotrophoblast level and mostly at the apical pole of the cytotrophoblast, where the cells need to fuse with the syncytial trophoblast. The last row showed IgG control, and they show no staining at all. So Ezrin localized in cytotrophoblast and a junction with the syncytial where it needs to fuse. Okay, so we have a complex Ezrin and Connexin-43, but we wanted to know if both proteins are directly linked together or if there is any other protein associated to that. So for this, we generate recombinant protein into bacteria by using P-destination vector and producing an Ezrin histidine tagged or Connexin with a GST tag. And we basically put all proteins together and did immunoprecipitation with the histidine tag. So we found Ezrin and histidine, of course. And in the tube where we had Connexin-43, we found Connexin to be pulled down with the histidine. In the same manner, if we did immunoprecipitation with the GST tag now, we're able to find Connexin-43 in the tubes where we had Connexin-43 as well as Ezrin, if we use Ezrin and histidine. But when we use only GST as a control, we're able to put on the GST, but there was no Ezrin associated to that. So this means that Connexin and Ezrin interact directly. We wanted next to study exactly the minimal amino acid sequence that could serve as an anchoring of Connexin and Ezrin. For this, we used the peptide array strategy. So what is peptide strategy? Let's say you have a full protein, such as Ezrin, for example, with the amino acid sequence. You will generate 20 amino acid peptides, and you will spot these peptides on the filter. And you will generate another peptide with one amino acid shift compared to the first one, and etc., in order to cover the whole sequence of the protein. And these peptides spotted will be spotted on the filter, and the filter will be probed with protein of interest, in our case the GST Connexin-43, and then you will reveal the interaction between Connexin and Ezrin by using an anti-GST HRP antibody. The other option is also to use a two-dimensional peptide array. So once you know the minimal amino acid sequence necessary to bind two proteins, you can use this minimal acid sequence and make a shift for each amino acid by the 20 other. And this will give you the information of which amino acid is important for the binding and which one can increase or decrease the binding. So the peptide array will decipher the minimal amino acid sequence necessary for the binding, and the two-dimensional peptide array will give you the essential amino acids for the binding. So this is what we did and spotted the complete sequence of Ezrin on the filter and probe with GST Connexin-43 or GST alone as a control. Each time you see the dark dots here, black dots, indicates you have interaction between the GST Connexin-43 with the peptide spotted. So then you can decipher which amino acid sequence is necessary for the binding. This is indicated in red. So we use this sequence and spot it again on the two-dimensional peptide array. This is on the right part of the slide. And here you can switch each peptide by one of the 20 other in order to see which peptide is essential for the binding, which one increase or decrease the binding. I indicated in the red squares two substitutions that show a decrease, a putative decrease in the association between Connexin-43 to Ezrin. And we wanted to actually support this data. So we generate a new peptide array in which we have the wild type sequence, and in the other one we also generate peptides that present these two substitutions. And we probe with GST Connexin-43, and we could see that with these two mutations, we decrease the binding of Connexin-43 to Ezrin. Okay, but what about in cell? So we generate a P-destination vector for GFP, Ezrin insensitive, presenting these two substitutions. And in that case, we are not able to reintroduce cell fusion. We have the presence of green aggregated cells. So this indicates that Ezrin, without the ability to bind Connexin-43, failed to rescue cell fusion. As I said earlier, gap junctions are really important for cell-to-cell communication. So in order to measure the communication between the cells, we did the Gap-FRAP assay, FRAP for fluorescence recovery after photobleaching. So how this works? We charge the cells with casein red orange M from Thermo Fisher. So this means that all the cells are stained, and you select one cell. And these cells, we use laser in order to bleach the whole fluorescence of the cells. And you will measure the recovery of the fluorescence over the time. This means that if the cells are able to communicate between each other, the recovery will come from the cells in the connected cells that are able to communicate. But if the cells are not able to communicate, then you have no recovery at all. So this will give you a sort of a spectrum on the right. We can measure the fluorescence recovery over the time of the bleached cells. And this is important to notice that this is live cell imaging, and we perform this on the spinning disk confocal microscopy. So if we treated the cells with scrambled Ezrin siRNA, we could see here is like there is a recovery of the fluorescence, indicating that the cells are able to communicate. And this is observed on the histogram here. We could see the diffusion rate constant increase. But when the cells were treated with Ezrin siRNA, we have a decrease in the communication. There is no communication or recovery from the fluorescence. If now we treated the cell with Ezrin siRNA and the plasmid GFP Ezrin siRNA and sensitive, in that case, we're able to recover trophoblast communication. But if now we use two constructs that produce a GFP Ezrin with a mutation in the PKA binding domain, or a GFP Ezrin siRNA sensitive that is not able to bind to Connexin-43, then we are not able to have a recovery of the fluorescence, indicating there is no communication between the cells. So this indicates that Ezrin modulates gap junction communication. So we wanted next to study from the connexin part and spotted on the peptide array the Connexin-43 sequence and probe the filter with the GST Ezrin or GST alone. Again, we can decipher the minimal amino acid sequence on Connexin-43 that is necessary to bind to Ezrin. And this sequence has been spotted for a two-dimensional peptide array. And you can see that if we make a mutation of this arginine by an acid aspartic here, by the red square, you could see there is a decrease of association between Ezrin and Connexin-43. So in order to support this data, we confirm again on new peptide array in which we have this single mutation, and you can see there is no association between Ezrin and Connexin-43. So what about in cells? So we treated trophoblast with Connexin-43 siRNA, and we are able to block the cell fusion. We have presence of aggregation compared to the scrambled siRNA. This is observed in the fusion index and the histogram here. But if we co-translate the cell with the Connexin-43 siRNA and a plasmid that will produce a GFP Connexin-43 siRNA and sensitive, we're able to reintroduce the cell fusion and the presence of a green syncytium. But if now we change the construct and produce a GFP Connexin-43 siRNA and sensitive that is not able to link to Ezrin, then we are not able to reintroduce cell fusion and we have the presence of the green aggregated cells. So this means that Connexin-43 without ability to bind Ezrin failed to rescue cell fusion. What about cell communication? So we use the same construct on the Gap-FRAP analysis. And here you could see that if the cells were treated with scrambled siRNA, we bleached the cells and you can see the recovery of the fluorescence, indicating cell communication. If the cells were treated with Connexin siRNA, we bleached the cells, and then after over the time there is no recovery of the fluorescence, indicating no communication. If now we treated the cell with Connexin siRNA and a plasmid encompassing a GFP Connexin-43 siRNA and sensitive, we can select the green cells, bleach the cells, then we have a recovery of the communication. And if we change the plasmid with GFP Connexin-43 siRNA and sensitive but not able to bind to Ezrin, we can select the green cells, bleach the cells, there is no recovery of the fluorescence. So this indicates that Connexin-43 without ability to bind Ezrin failed to rescue gap junction communication. Like I said earlier, Connexin-43 can be phosphorylated by many kinases and PKA also. Here is a representation of the C-terminal part of the Connexin-43. I stand in red all the serines that can be phosphorylated. Interestingly, in the extreme C-terminal part, you could see 6 serines that can be phosphorylated. Until now, Many people found that only the serine 364 were phosphorylated by PKA, but what about the others? I indicated also in the arginine in pink here, that is the amino acid necessary for the association with Ezrin. This is interesting to notice that these 6 serines are in the same area of binding with Ezrin. So in order to figure out if PKA can phosphorylate directly Connexin-43, we culture the cells with PKA inhibitors that we call PKI, or the scrambled peptide, and stimulate the cells with cyclic AMP or not. And what you can see in the cyclic AMP stimulated cells, there is an increase in phosphorylation of Connexin-43 in the cells treated with scrambled PKI that is reduced with PKI peptide. We also observe here the phospho-PKA substrate that more or less show the activity of PKA, and you can see that in presence of cyclic AMP, we increase the activity of PKA that is reduced in presence of PKI. So this indicates that when we make the ratio of phospho-Connexin-43 over the total Connexin-43, PKA seems to phosphorylate Connexin-43 in human trophoblasts. What about gap junction communication? So here the cells were treated with beta-GA, which is a classic inhibitor for gap junction communication that block the communication compared to the control. But here the cells were treated with scrambled PKI. We also, and stimulate with ACG, were also able to observe a communication between the cells that is completely blocked or abolished by treating the cells with PKI. So PKA mediates gap junction communication in human trophoblasts. As I said earlier, the anchoring of PKA through AKAPs, in our case Ezrin, can phosphorylate directly a target and give a biological effect. If we use anchoring disrupted peptides, you lose the biological effect due to the distance of the catalytic subunit that does not phosphorylate the target anymore. And this is what we did here. We treated the cells with RIAD or super AKAPs. If you remember, this is PKA type 1 and PKA type 2 specific anchoring disrupting peptides, or both of them, and stimulate the cells with ACG. And what you could see is when the cells were treated with this peptide, we completely decreased the phosphorylation of Connexin-43 compared to the scramble control. So this indicates that the anchoring of PKA is necessary to phosphorylate Connexin-43 in human trophoblasts. What about gap junction communication? Here again, this is Gap-FRAP analysis. So the cells, in cells treated with scrambled RIAD or scrambled super AKAPs, you could see there is, we can measure the intercellular communication with the recovery of the fluorescence in the cells. But if the cells were treated with either RIAD or super AKAPs alone, then you can observe no recovery at all and a decrease in intercellular communication. So this indicates and supports the preliminary data that we found with Ezrin, showing that the anchoring of PKA facilitates gap junction communication in human trophoblasts. Okay, but then we wanted to exactly know which serines would be phosphorylated on Connexin-43. So for that, we generate Phospho-Peptide Array. And for this, we used the Connexin-43 phospho-sequence that we thought to be PKA specific, and introduce single or multiple combination of serine substitution with alanine and spotted this peptide on filter. And then the filter were subject to PKA phosphotransferase assay. So each time the PKA can phosphorylate the peptides, you can measure by the radioactivity, or if there is no phosphorylation, you will see no phosphorylation at all. So this will delineate the PKA phosphorylation site in Connexin-43. And this is what we observe here on the left part. On the right part is the quantitative analysis of the PKA activation. So what we could see is that the histogram, the first histogram, show the phosphorylation level of a consensus PKA phosphorylation sequence. This is in black. Or gray, you will see a CREB sequence phosphorylated by PKA. So both of them are highly phosphorylated by PKA. And as negative control, we use the consensus CK1D phosphorylation sequence that show no phosphorylation at all by PKA. And we use as a control, of course, the wild-type sequence of Connexin-43 that show in blue a phosphorylation by PKA. Interestingly, if we introduce one single mutation each time for serine 369 or serine 373, we have a decrease in the phosphorylation level compared to the other phosphosites. So this is what we observe here, the data on the Phospho-Peptide Array. Original data on the left and on the right correspond to the analysis of this, the PK activity. So we generate peptides with consensus PK phosphorylation site. This is the black histogram. And in gray histogram, you will see the CREB with PK phosphorylation site sequence. And both are really phosphorylated by PK. As a negative control, we use a consensus CK1D phosphorylation sequence that is not phosphorylated by PKA. Well, if you use the wild-type sequence of Connexin-43, which is the blue one, you can see a phosphorylation of the sequence by PK, indicating that PK can phosphorylate that sequence. If not in that sequence, we include one substitution in serine 369 or serine 373 by an alanine, we decrease the level of PK phosphorylation. But more interestingly, if we include a double mutation substitution of these two amino acids, serine 369 and serine 373 with an alanine, we completely reduce the PK phosphorylation. So this indicates that, at least on the filter, PK phosphorylates Connexin-43 on serine 369 and 373. To support this data, we decided to make phospho-proteomic analysis. We have cells unstimulated or stimulated with cyclic AMP in order to activate the cyclic AMP PK signaling, and then realize immunoprecipitation with DNABits to enrich all the Connexin-43, make trypsination, and analyze the peptide on LTQ arbitrage to identify the phosphosite. And here are listed all the peptides which we found or on the area that we're interested in. And what we found is, like, in the basal condition, many of the serines has been basal already phosphorylated, such as 364, 55, 65, 68, and 69. But the most interesting, like, upon acute stimulation of the cyclic AMP, one peptide came out is the phosphorylation on serine 373 or serine 372. So based on these two analyses, Phospho-Peptide Array and the Phospho-Proteomic analysis, we can conclude that the phosphorylation of Connexin-43 on serine 373 is actually due to an activation of cyclic AMP. What about this construct in cells? So we generate new destination vector that will produce a GFP Connexin-43 siRNA insensitive, not able to link to Ezrin, with substitution that mimic either a non-phosphorylated form of the serine, which is a substitution with alanine on serine 369 or 373, or a phosphomimicking variant by switching this serine with an acid aspartic with a D, and then treated the cell, transfected the cells with either scramble siRNA or Connexin-43 siRNA and the plasmid. And what we could see by doing this is the cells that were transfected with a scramble siRNA present syncytial trophoblast or fusion, while the Connexin-43 siRNA transfected cells present aggregation, aggregate cells with low fusion index. And if now we treated the cells with siRNA plus a GFP Connexin-43 siRNA insensitive, as earlier, we're able to introduce cell fusion, presence of yellow syncytium. And if now we make a mutation substitution into this Connexin-43 in order to abolish the binding with Ezrin, we are not able to introduce cell fusion. And what about the phospho-mimicking variants? Each time the constructs were transfected with construction that lack the phosphorylation on serine 369 and or 373, we are not able to reintroduce cell fusion presence of aggregated yellow cells. But each time the construct presents a phosphorylation variant with acid aspartic instead of the serine on 373 or 369, we're then able to reintroduce cell fusion. So this indicates that the phosphorylation on serine 369 and serine 373 is very important to control the cell fusion. So we wanted now to analyze this intercellular communication. And for this, we didn't perform Gap-FRAP analysis before. We performed Gap-FLIP, fluorescence loss in photobleaching. So again, this is live cell imaging, and we charge the cell with calcine red, orange, AM, and we select, instead of bleaching a complete cells as before, we only select a small area of the cells, and we'll repeat a laser beam in order to bleach over the time the cells. And if the cells are able to communicate between each other, we will also bleach the connected cells. So you can measure over the time the fluorescence loss of the bleached cells, but also the connected cells. And this can give you which cells are actually communicate between each other. So this is what we did here. And we transfected the cells with GFP control to measure the intercellular communication. So we give you the basal communication. We did an overexpression of GFP connexin SRNA sensitive that show you a high communication. And if we replace this construct with a construct that is not linked to Ezrin, we are not able to communicate anymore. But more interestingly, if we look and focus on the phosphomimicking variants of Connexin-43, each time the serine 369 or 373 were not phosphorylated by presenting an alanine substitution on the serine, we are not able to reintroduce intercellular communication. While when this construct show phosphorylation mimicking variants on serine 369 or 373, then we are able to introduce intercellular communication. So the take-home messages is the cyclic AMP PKA signaling controls human trophoblast fusion. Ezrin anchors PKA and controls cell fusion in human placenta. Ezrin forms a signaling molecules together with PKA and Connexin-43. And Ezrin bonds to Connexin-43, controls trophoblast fusion and intercellular communication. PKA anchored to Ezrin phosphorylate directly Connexin-43 on serine 369 and 373, and these phosphorylation sites promote cell fusion and intercellular communication. So, in conclusion, you will find gap junction here in the basal state with Ezrin linked to Connexin-43 and PKA associated to Ezrin. Once you will have an increase in cyclic AMP concentration, you will release the CC unit that will phosphorylate Connexin-43 on serine 369 and 373, which will open the gate and transfer fusionic molecules or signals that will allow to start the fusion process. I would like to thank many people that give me the opportunity to make this work. Of course, I want to thank the people from the INSERM 767 and more precisely Pascal Jabot and Fatima Ferra. We did a large part of that work, but also the EMBL Nordic in Norway, and more precisely the Tasken group, Kjetil Tasken and Alexandra Dukic, a PhD student, had been done a large part of the work as well, and the TIL group for the mass spec analysis. I want to thank the Institut Curie, Vincent Frézy and Ludovic Lecomte for all the live cell imaging, and of course I want to thank at Thermo Fisher Scientific, Olivier Eder and Jérôme Selsati that give me the opportunity to present all this work here today. Thank you so much.
Thank you, Dr. Pidoux, for that informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question drop-down box located on the far left of your presentation window. Type your question into the box that appears on your screen and click the Send button. We'll answer as many of your questions as we have time for. Now let's get started. Our first question is, why did you change from Gap-FRAP to Gap-FLIP analysis?
Okay, thank you. While you are doing gap junction intercellular communication, you want to monitor kinetics of communication between connected cells, right? And in both cases, you can use FRAP analysis, which is fluorescence recovery after photobleaching, or FLIP analysis, which is fluorescence loss in photobleaching. So you have live cells that are charged with calcium dye, so that it became fluorescent. And in FRAP analysis, the entire fluorescence of the target cells will be bleached in one run of a laser beam, and you measure over the time the fluorescence recovery. This fluorescence comes from connected cells to the target cells. So this works perfectly fine, but this is a huge amount of work for analysis and to identify, after all, how many cells are actually truly connected to the target cells. And if they're connected, then all the cells are connected in a similar manner, meaning if they are connected, yes, no, and are they connected giving them more fluorescence or not. Then we decide to switch to another analysis, which is the FLIP assay. This is something completely different. In this case, you choose a targeted cells, and you will bleach a small area of these selected cells, and you will bleach over the time in the same area, over and over. It's repeated. So you will measure, you will see the fluorescence loss of that cells. This is completely the opposite of the FRAP. And then, over you bleach these cells, the neighbor, which are connected to each other, if they are communicated to the targeted cells, then they will lose as well the fluorescence. And then you will measure, in that case, the fluorescence loss not on the targeted cells, but directly on the connected cells. So then it becomes more accurate to measure these kinetics. And then you don't have really to worry about how much and how many cells are directly connected to the targeted cells. This actually helps for the analysis.
All right, and can you explain how you did not find phosphorylation of Connexin-43 at serine 364, while until now it has been described to be the main PKA phosphosite?
Yeah, to be honest, well, we don't know exactly. It could be many possibilities. So we haven't found this serine to be phosphorylated by two main experiments, which are mass spectrometry analysis. So in mass spectrometry, we found this serine directly phosphorylated in basal state. And by peptide array, phospho-peptide array, we haven't found a direct phosphorylation of that site by PKA compared to two other sites. So what we have to keep in mind is that when we did peptide array, this is peptides that are synthesized and then spotted on the filter, and they actually lack the 3D structure that's probably important in real life. So we could imagine that on the filter, the 3D structure is not on the filter. The 3D structure is not correctly folded, and then the PK cannot phosphorylate directly these phosphosites compared to two other. And that could be an explanation why we didn't find it. Or, as I said earlier, that we haven't found PKA or in basal state PKA after stimulation of the PKA signaling activation to phosphorylate this site. This could be due to, or you have at the basal a tonic activity of the kinase activity that phosphorylates any of this phosphorylation, this site, and this is not actually observed on the peptide array due to the 3D structures, or we cannot exclude that PKA can activate other
signaling molecule down effectors that could phosphorylate indirectly these phosphosides. Or, of course, we cannot exclude or rule out that PKA does not phosphorylate at all this serine.
All right, and looks like we're going to have one more question. Is this complex specific to human placenta?
Yeah, and this is a good question. Actually, we have been working on human placenta for over 15 years, and we actually identified this complex PKA Ezrin Connexin-43 as a key regulator for gap junction cellular communication that leads to the placenta formation that is necessary for fetal growth and in all the pregnancy, actually. But we actually developed some collaboration, and we are still working on other complexes in other cell type, and we found that this complex is also present in T cells, for instance. That's very important for the T regulation, Treg cells in the immune system, and also in the liver cells, where the complex is also important for the communication between the cells. So we cannot exclude that actually this complex is probably ubiquitous and could be present in all cells that need to communicate between each other, but in which the cyclic AMP PK activation actually play a key role.
All right. Well, I would like to once again thank Dr. Pidoux for his presentation. I would also like to thank Thermo Fisher Scientific for making today's educational webcast possible. And before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through September of 2019. Please share with your colleagues who may have missed today's live event. That's all for now. Thank you for joining us, and we hope to see you again soon. Goodbye.